Ancient Greece, where the foundations of modern medicine were laid. Here, Hippocrates practiced medicine in the belief that all illness had a rational explanation. Aristotle wrote of the anatomy of man and animal. And Dioscorides was said to travel the world in search of medicinal substances. These advances were just some of the many attempts to find explanations for and cures to human disease. Today, that search continues at SAIC Frederick. A wholly owned subsidiary of the Science Applications International Corporation, SAIC Frederick is the operations and technical support contractor for the National Cancer Institute at Frederick, Maryland. The mission of the men and women of SAIC Frederick is to provide support to the National Cancer Institute and others within the National Institutes of Health in the search for the causes and cures for cancer and infectious disease. Scientific, technical, and administrative specialists work together to fulfill these goals. SAIC has a well-known reputation as a science and technology company helping to invent the future. And at SAIC Frederick, we believe the future holds the cure. Nestled against Maryland's Catoctin Mountains, SAIC Frederick sits on nearly 70 acres. This strategic location in the city of Frederick places them within one of the most active biocorridors in the U.S. and with easy access to the cities of Washington, D.C. and Baltimore. This double-stranded helix of nucleotides has long been considered the key to unlocking the nature of humanity. And the sequencing of the human genes that make up DNA was a massive undertaking. Now, born from genomics, is proteomics, a new generation of science that studies proteins, which are the actual products of those genes and the building blocks of life for all biological systems. These proteins carry out functions vital to energy production, communication, survival, and growth. In a joint effort with their NCI colleagues in both Frederick and Bethesda, SAIC Frederick scientists are working with the Food and Drug Administration to pioneer a technique of proteome pattern recognition for ovarian cancer. Difficult to diagnose early, ovarian cancer has a survival rate greater than 90% if the disease is discovered and treated in its early stages. With a highly sensitive mass spectrometer, investigators are able to capture snapshots of a complex and unique array of proteins in the blood, measure their mass, and identify them. Tens of thousands of proteins can be expressed by a variety of genes at any one time. Rather than isolate each protein individually, a time-consuming process, investigators use a high-throughput divide-and-conquer strategy. Big proteins are divided into smaller portions known as peptides. Like fingerprints at a crime scene, patterns of peptides reveal the proteins they come from. High-throughput identification quickly provides investigators with a list of over 1,000 proteins and enables them to establish distinct proteomic patterns. The protein patterns in an ovarian cancer patient's blood sample are compared to samples from individuals without cancer. With this information, investigators hope to create a tool for pattern recognition that enables them to diagnose and gauge how far the cancer has advanced. The earlier the diagnosis, the better the chances are for recovery. Comparable concepts are already underway for prostate, breast, and lung cancer. Once these techniques are validated and approved by the FDA, they may be licensed to a manufacturer for commercial delivery. On the surface, scientists in these labs may appear to be performing routine established procedures. However, here in the Basic Sciences Laboratories at SAIC Frederick, concepts and state-of-the-art methodologies are developed with the hope of finding later applications to cancer and infectious disease. Some projects identify unique molecular targets in cancer cells that can be attacked by new drugs or by the body's own immune system. For example, natural killer, or NK cells, have been identified as cells that are critical regulators of the body's ability to recognize cancers or infectious agents and initiate their destruction. Some people have NK cells that are more effective in attacking diseases than others, and this is due in part to differences in genetic makeup. Investigators have found a link between this genetic variation and the incidence of cervical cancer and with progression to AIDS after HIV-1 infection. With this discovery, researchers are attempting to determine the functional basis for the genetic findings, and they can investigate the role of these genetic variants in resistance to other cancers. Finally, they hope to use this information to generate new strategies for tackling cancer. For example, some cancers express unique molecules that can serve as targets for treatment strategies. Follicular lymphoma is a cancer of some white blood cells known as B lymphocytes. Sometimes, as B lymphocytes develop in the follicles of the lymph nodes, something goes awry and they reproduce uncontrollably, resulting in a tumor. By understanding the body's own immune system, scientists at SAIC Frederick are developing new molecules that can recognize and destroy these cancer cells. After analyzing an original biopsy and identifying the right candidates, large quantities of these unique reagents are made, joined to other specialized carrier molecules to create a vaccine that can be injected into the patient. Follicular lymphoma is a slow-growing tumor which can take years to show itself, and even after discovery and chemotherapy treatments, often recurs. Clinical trials of this new vaccine are underway with the goal of preventing such recurrence after remission. In the past 20 years, the number of deaths attributed to the human immunodeficiency virus, the cause of AIDS, is nothing less than devastating. Worldwide, millions of lives have been claimed, and tens of millions more currently suffer from the terrible effects of HIV. HIV causes not only AIDS, but is also associated with many types of cancer. With the expertise and a technical infrastructure in place, the National Cancer Institute assumed an early leadership role in combating this difficult disease and made headlines by making key contributions to the first AIDS tests to protect the nation's blood supply. NCI scientists then played a key role in developing the earliest effective treatments for this disease. The search for vaccines and effective drug therapies continues today in the AIDS vaccine program. Using basic and applied research, investigators study how HIV causes disease and are developing classes of possible vaccines to prevent infection or the consequences of an infection. Studies have led them to a new approach using innovative vaccine strategies that might be used by physicians to stimulate the immune system and prevent HIV infection. Other investigators are studying how the HIV virus constantly changes itself to become resistant to new drugs. These scientists seek to find a critical Achilles heel in the virus so that much more effective treatments can be developed. As new targets for prevention and treatment of cancer or AIDS are identified, promising drugs against these targets must be produced. The Biopharmaceutical Development Program, or BDP, acts as a highly specialized biopharmaceutical company that produces the small amounts of new drugs needed for the earliest clinical trials. In these clinical trials, the National Cancer Institute assumes the initial financial risks inherent in drug development to expedite bringing new treatments to a point where commercial pharmaceutical companies will continue the development process. In these fermentation tanks, the BDP grows unique genetically altered bacteria that produce the materials to be tested as possible vaccines or drugs. One project involves the creation of tumor-targeting monoclonal antibodies that can be injected into cancer patients to treat or detect cancers. For example, the polio virus, an old enemy, is now an ally in fighting glioblastoma, the most common and aggressive type of primary brain tumor. The polio virus has now been modified so that rather than killing healthy cells, it attacks the malignant cells that form the deadly tumor. BDP scientists and their university collaborators are working to deliver this exciting technology to physicians and their patients. The BDP team's collective expertise covers the development, manufacturing, testing, and quality assurance of new biopharmaceuticals. In the end, products are brought into the highly restricted clean room for packaging, where they undergo a final purification process. Working with the FDA, the BDP staff delivers products for clinical and efficacy trials. Afterwards, they may go to commercial pharmaceutical companies for further development toward the goal of eventually being approved for use in treatment. The research at SAIC Frederick is supported by a variety of different high-technology development groups. Here in the Protein Expression Laboratory, DNA is cloned to make mass quantities of protein molecules for lab work. Large quantities of cells are produced to provide raw materials for protein purification. These crude protein mixtures are then separated by sophisticated machines into pure component fractions. Advanced information technology is essential to the work at SAIC Frederick, which is supported by the Advanced Biomedical Computing Center. The ABCC houses an impressive array of high-performance computers, 64-node servers, and a robotic tape cycle that stores terabytes of information. The diverse databases of the National Cancer Institute are brought together for ease of use and maximum accessibility, implementing NCI's Center for Bioinformatics Software via high-performance platforms. The Computer Center provides support for all investigative efforts, from molecular modeling to the design of clinical trials. These databases link the campus and academic institutions around the world. Seeking the healing powers of natural products, from the ocean floor to the bark of trees in tropical forests, is the job of the Natural Products Support Group of the Developmental Therapeutics Program. Labs at NCI search relentlessly for new molecules that may kill cancer, inhibit viruses, or prevent growth of harmful bacteria or fungi. They obtain plant, marine, and fungal specimens from all over the world and bring them to this research group for processing. Since 1987, over 180,000 extracts from specimens have been prepared, from which promising compounds with therapeutic qualities are purified. These samples are meticulously weighed, barcoded, and stored for use in NCI labs, as well as in academic and biopharmaceutical labs around the world. Breaking up these extracts into smaller fractions is a process called high-throughput dereplication, which identifies biologically active material. With today's modern laboratory techniques, all 180,000 samples can be utilized and tested by other NCI labs within a matter of weeks. Specialized storage space is critical for large-scale scientific operations. National Cancer Institute repositories store millions of samples, ranging from natural and synthetic compounds to drugs and clinical materials. This vast inventory is vital to the research effort and uses a worldwide web-based system so scientists can easily acquire samples for evaluation. In the lab of molecular technology, researchers can automatically sequence genes. Using PCR machines and robotics that can operate 24 hours a day, the staff prints thousands of samples onto microscope slides that can be used on campus and by researchers across the country. While the advanced custom-made machinery speeds this process, the lab is also used as a site for evaluating 21st century biotech equipment. The labs of SAIC Frederick at the National Cancer Institute house the very latest technologies to assist in the search for the cures to disease. While a tried and true scanning electron microscope and transmission electron microscope are available, the image analysis laboratory also boasts two confocal microscopes. Inverted so a lens never comes in contact with a plate, the microscope's scanning motors quickly move laser beams over a cancer cell specimen. Using a technique known as fluorescent microscopy, laser light penetrates cell tissue and allows the user to excite molecules. With a conventional microscope, only one layer of cells can be seen. But with confocal technology, multiple layers of tissue cells can be rendered in startling 3D. Several nuclear magnetic resonance spectrometers are also on campus. Researchers place samples into the powerful magnet, which reveals the actual arrangement of molecules and provides a view of proteins at the atomic level. SAIC Frederick scientists also have full use of the services of the Scientific Library, a wonderful resource and a quiet place to read up on the latest scientific publications. More than 800 journal titles can be found on its shelves, and there are online resources, books, employee reference materials, and hands-on training in the microcomputer lab. A multimedia teaching auditorium and conference center play host to seminars and the presentation of research papers. A variety of effective media tools are available in the Office of Scientific Publications, Graphics, and Media. Graphic artists create print materials such as posters for presentations and assist laboratories in explaining complex processes in full color. Still photography, digital imaging, and the latest in digital video production are also available, as well as manuscript writing, editing, and printing. Providing a respite from the glow of computer screens and the hum of scientific labs, the National Cancer Institute's Spring Research Festival is held every year under a mammoth tent erected especially for the event. Sponsored in conjunction with the U.S. Army Medical Research and Materiel Command, the gathering allows both scientists and citizens alike to get acquainted with the latest cancer research. Here, resident scientists and technicians describe their work. Also included are a health and safety exposition and commercial exhibits of the latest scientific equipment and technologies. The benefits of working at SAIC Frederick go beyond being part of this important mission to cure cancer and infectious diseases. Employees enjoy a wealth of housing choices in the Baltimore-Washington corridor. They might choose to live in Frederick, the second largest city in Maryland. Steeped in history, it is now home to more than 50,000 people. Francis Scott Key once lived and practiced law here, and beautiful parks, pools, and fairgrounds fill out the historic streetscapes. Harry Grove Stadium fields a minor league team of the Baltimore Orioles. Washington, D.C., Baltimore, Virginia, West Virginia, and Pennsylvania are all within commuting distance. And the National Institutes of Health in Bethesda is only 35 miles away. Fishing, hiking, and other recreational activities abound, and the Atlantic Ocean is a mere 3-hour journey. Convenient on-site daycare is available to SAIC Frederick employees, as well as attractive health coverage, life insurance, and retirement plans. Prepaid tuition assistance is available. The area offers excellent public and private grade schools for children, and institutions of higher education such as Frederick's Hood College, the University of Maryland, Georgetown, and the Johns Hopkins University are all a short drive away. The greatest attraction of SAIC Frederick, however, is the dedicated staff from all around the globe. Diversity is part of life here on campus, from the administrators and scientists to the students and postdoctoral fellows gaining valuable experience as the next generation of cancer scientists and researchers. Employment opportunities are equally diverse. As the operations and technical support contractor, SAIC Frederick's workforce includes a broad array of expertise in numerous support capacities, ranging from facilities maintenance to business and logistics. Cancer is one of humankind's oldest enemies, a rubric for over 200 health-related problems. The ancient Greeks took some of the first steps toward improving human health almost 3,000 years ago. The people of SAIC Frederick at the National Cancer Institute continue to accelerate the progress of scientific research for cancer patients and people at risk. SAIC Frederick, where the future holds the cure. For more information on career opportunities at SAIC Frederick, please visit our website